In this video, we'll be looking at this squirrel eating some corn. Also, we'll be doing some multiple choice questions on the topic of prokaryotic and eukaryotic cells. Your teacher will definitely use some of these questions, so this is a good place to come to prepare for a test. Always practice what the exam will be, and that's doing questions. So let's get into it. First, I recommend you try the question by yourself because maybe you know it already. So let's get into it. And by the way, these are real past paper questions, so I screenshot them, so that's why the questions are in no real order. So what distinguishes prokaryotic cells and eukaryotic cells? So, A, cell wall, plasma membrane. No, this is not the case because although, yes, prokaryotic cells does have a cell wall, so does some eukaryotic cells. We know that eukaryotic cells are animal and plant cells. So here is an animal and here is a plant cell. Although not all eukaryotic cells have a cell wall, plant cells do, and they are eukaryotic. So we can't distinguish prokaryotic cells and eukaryotic cells based on cell wall because plant cells does have a cell wall. So does prokaryotic cells. So therefore, this is not a good way to distinguish because eukaryotic cells do not only have plasma membranes, they also have cell walls. So definitely not A. What about B? Cell structure not compartmentalized and cell structure compartmentalized. Now, what do they mean by compartmentalized? What they mean by this is the cell has compartments in it, and this means organelles. So for example, let's look at this eukaryotic cell. We can see inside the cell, there are compartments. So for example, there's a cell wall surrounding this mitochondria, and there's a cell wall surrounding this rough endoplasmic reticulum. There's a cell wall surrounding the nucleus. Everything is compartmentalized. There is different compartments, a compartment for the nucleus, a compartment for the mitochondria, a compartment for this, and a compartment for that. Whereas prokaryotic cells just has everything blobbed inside the cell. There's no compartments. There's nothing separating the DNA from the cytoplasm. There's nothing separating enzymes from the cytoplasm. Everything is just a blob. Okay, so that's what they mean by compartmentalized. So yes, this is true. Prokaryotic cells have no compartmentalization, whereas eukaryotic cells do. So we know it's B. Let's just eliminate these two options. So prokaryotic cells has a smooth endoplasmic reticulum, whereas eukaryotic cells has a rough endoplasmic reticulum. Again, this is not true. Eukaryotic cells have both smooth and rough. Remember, rough is when it has ribosomes on it, and smooth is when it's without ribosomes. And prokaryotic cells don't have any organelles, basically. So this statement is simply not true. Okay. Ribosomes and no ribosomes. So prokaryotic cells and eukaryotic cells have ribosomes. We can see here, there's some ribosomes, here's some ribosomes in the plant cell, there's some ribosomes in the prokaryotic cells. So both prokaryotic and eukaryotic cells do have ribosomes. So this one is not a way to distinguish between them. So therefore the answer is B. Let's go on to the next one. Give it a go yourself. So what we have here, this image represents E. coli. E. coli is a type of bacteria, and we know bacteria are prokaryotes. So this image they're showing here is pretty much a prokaryote, a bacteria, a specific species of bacteria called E. coli. Now, what is the function of structure X? So they're pointing at these pili, right? We know when we label a prokaryote, we can label these little lines as pili, and they are different from flagellas. Flagellas cause movement, whereas pili don't cause movement. They cause attachment and communication. They have various functions, but the most important one is attachments. It's like little hands grabbing onto different surfaces. Okay, so they're asking you, what is the function of this structure X? Active transport? No. Active transport is when things are moved in and out of the cell. These pili don't do that. All they do is attachment. So attachment, the answer would be B. We know little pumps in the membrane, in the plasma membrane, called protein pumps, can pump things in and out, but not these, not the pili. How about binary fission? This is definitely not an option because binary fission is a way of splitting, so it's not really a structure. Cell respiration. So this thing does not do cell splitting, okay? It does not do binary fission. Cell respiration, no. Cell respiration would happen inside the cell, okay? Normally in a eukaryotic cell, it happens in the mitochondria, in the specific compartment of the mitochondria. But we know prokaryotic cells do not have compartments, so it just happens randomly inside the cytoplasm. There's no specific compartment for it. Okay, next one. Again, a lot of the questions, as you do questions from the IB, if you do enough, you notice they repeat quite a lot. So after a while, doing the questions just becomes pretty straightforward. So that's the key to succeeding, to be honest. So what distinguishes prokaryotic cells from eukaryotic cells? So you can see it's a pretty damn similar kind of question to this one. Okay, so let's see what's different. They're saying no plasma membrane, plasma membrane. Again, we know they both have plasma membranes. Okay, so this is not a way of distinguishing between them. 80S ribosomes, 70S ribosomes. So this is not true. Actually, eukaryotic cells have 80S ribosomes and prokaryotic cells have 70S, so they swapped them around. And remember, S is pretty much just a unit of measurement. So for example, if I say I am 2 meters tall, the unit of measurement is meters, right? So just like meters is a unit of measurement, S is a unique measure of unit of measurement for ribosomes. It's a way of measuring the size and various properties of a ribosome, okay? And eukaryotic cells have 80S ribosomes, whereas prokaryotic cells have 70S. So this one is not true. Golgi apparatus and mitochondria. Prokaryotic cells don't have organelles, so they don't even have a Golgi apparatus. And it's true, eukaryotic cells do have mitochondria, but since this one is not true, this answer can't be right. Last one. We know then, therefore the last one must be right just because we eliminated these, but we'll double check. So no internal membrane compartments and internal membrane compartments. Doesn't this sound exactly like the question we just did? This is another fancy way of saying no compartmentalization, which is what we literally just saw, okay? And we know prokaryotic cells are not compartmentalized. Everything is just blobbed in the cell. And we know eukaryotic cells do have internal membrane compartments. The cell is organized into different organelles with different walls and compartments. Okay, so this one's pretty straightforward, just like the other one. It's D. Now, I hate these kind of questions because this next one, because I really struggle with them, but hopefully I can help you with this. The image, let me look at the question first and I'll let you give it a go yourself. So this image shows an electron micrograph of mesophyll cells. Okay, so I said in my other video that in order to be able to see organelles inside a cell, so in order to be able to see the mitochondria, the nucleus and all this, you need an electron microscope. A light microscope will only show you the cell surrounding and the nucleus. It won't show you organelles. So you need an electron microscope in order to be able to see organelles, okay? A light microscope is not enough. So we can then know that when they're talking about an electron microscope, we can think we'll be able to see organelles. Okay, what is a mesophyll cell? You need to remember this. This is the only plant cell they want you to care about. It's called a palisade mesophyll cell. Just know when you see this name, it means plant cell. Therefore, you can immediately think back to plant cell structure and then can take into consideration the picture. So we can see here a bunch of cells, right? 1 cell, 2 cell, 3 cell, but it looks really weird, doesn't it? What's this massive white space? So remember, in the video when I talked about plant cells, I said plant cells are unique because they have massive vacuoles. So in this picture I showed here, it's actually quite small, right? Because here's a real picture of a plant cell, and we can see they have humongous vacuoles. Huge. Now, what are these little things then by the side? So we can see this vacuole is full of water, and as it fills up with water, it squeezes all the organelles to the side of the cell till it touches the cell wall. Okay, so what are these little organelles? If you were to look at this picture, what would you think it is? It could be a mitochondria, it could be maybe a nucleus, it could be or a chloroplast, it could be any of these, right? So what makes you think? What makes you decide what it is? So we know they give the options cytoplasm, mitochondria, nucleus, chloroplast. We can eliminate cytoplasm because they're pointing at this structure, and cytoplasm is not an organelle. It's the It's the entire blue space in this picture. It's what the organelles is dissolved in. So, not a cytoplasm. Could it be a mitochondria? I would say it's not a mitochondria because a mitochondria has a pretty distinct structure. When you think about a mitochondria, you think of a thing with these little lines on it like this, and they're typically a bit smaller than this structure. So you can see the lines on this one isn't characteristic of a mitochondria because they're not like this, up and down, up and down. They're more throughout. They're more like a long line throughout the whole cell, a bunch of long lines like this. So when you see a bunch of really long lines extending throughout the organelle like this, you should think it's a chloroplast, okay? So this is a really tricky question because you never know, is a mitochondria a chloroplast? Because this plant cell has both, mitochondria and chloroplast. But you should know it's a chloroplast based off experience. Just look at a few pictures of chloroplasts and you'll see they all look unique. They all look like they have several long lines from one point to the other point. Whereas a mitochondria doesn't have lines from one point to the other point. It rather has a wiggly line across like this, okay? But it's hard to see, so I do understand if you find this one hard. It's not a nucleus because a nucleus is round. These are not exactly round shape, as you can notice. But it's a tough question, to be honest. Okay, let's move on to the last one here. Give it a go by yourself. What is the structure labeled X in the electron micrograph of the rat cell? So when I first did this question, I was like, do they mean like what specific thing like here? No, what this question means is what is this whole structure? What is this whole structure? Again, we're talking about an electron micrograph, which means an electron microscope was used. So we can see really tiny things. We can see organelles. We can see all that. In this case, it looked like they zoomed in to a complete organelle. So this is not a cell. This is an organelle inside a cell. It really looks like they zoomed in to an organelle like this, for example. They zoomed in even more to be able to look just at an organelle. That's what this picture is about. So, what organelle would you say this is? Remember how I just said, when there's lines wiggling, waggling like this throughout the organelle, you should think mitochondria. Yes, this is a mitochondria. Whereas a chloroplast would rather have lines going from one point through all the way to the other point, kind of like this. Whereas mitochondria has them wiggling, waggling like this. Okay, so this would be a mitochondria, whereas this one above is a chloroplast. So that's one key way of being able to tell. And anyways, besides knowing that, you should have known it's a mitochondria because it's a rat liver cell, not a plant cell. So this cannot be a mitochondria. I mean, this cannot be a chloroplast because animal cells do not have chloroplasts, remember? So therefore, it's a mitochondria. It's not a ribosome because a ribosome would be like really tiny dots. Okay, this is not a tiny dot. A ribosome would look like this. It would be, if this is a mitochondria, this is a ribosome. It's a really small dot. You can barely see it properly with a mitochondria. You almost can't see it with an electron micrograph. Okay, a lysosome is probably these. It's a little bubble. It doesn't have lines in it like the mitochondria does. And again, a nucleus is really large compared to this, and it's round. Whereas this structure, a mitochondria, is not round. So I hope these questions helped a little bit. Some of them, I agree, they are tricky. It's really hard to see, especially if you're just learning about biology and you're not a teacher or anything like that, because you don't have experience. But I would just recommend Google a bunch of pictures of mitochondria and just get used to how they look like. Just get some experience in, because otherwise you get surprised when you come to the test and you've never looked at an electron micrograph of any of the organelles. So just get used to it. I'd say just build up the experience of how it looks like, and then it comes really easy when you get to a test.